So it's really great to be here. Chicago is first time for me. So everything was smooth. It's so well organized. It's very impressive. I had a Japanese chocolate. So I'm sure this is the coincidence, but I feel it's very sweet. So I'm going to talk about gene regulation of senescence, but I mostly focus on the 3D organization of chromatin. So the pointer, I'll use this pointer. Okay. So I just started with this acknowledgement. So today's talk mainly driven by my postdoc. She's a mathematician, Joanna Olan, here. She's in the audience. Thank you very much for inviting both of us again. And then it says great cross collaboration between the mathematics and then the genomics and cell biology and so on. Tetsuya is very good at technology application, and lots of experiments are so tedious. There's so many steps you can make mistakes, and then only high quality genomic data pass through Joanna's filter. It's that one of the most difficult parts to complete the experiment. Masako, actually my partner, I'm going to share the chocolate with her when I'm back. She's a cell biologist like me, and then organize everything. And the two previous students, Alet, Masami, generated quite a few data sets. So what's senescence? We still don't know much about senescence. Everybody has a different definition, right? Our definition largely based on epigenomics or 3D epigenetics. But in terms of biology, so like apoptosis, lots of diverse stimuli can induce senescence. But we all usually talk more on like two aspects of senescence, inside and outside. We call the cell autonomous aspect and the cell non-autonomous aspect. Like here you see the terminally arrest, right? Stable cell cycle arrest. This is probably the most critical one. Without this one, there's no senescence, right? And reduced plasticity. This is still ongoing project. We are now working on it, but I'm sure this is reasonably good assumption. And together with those two aspects, this is autonomous tumor suppressor. Right? We used to call senescence as a tumor suppressor, but we have to be careful. It has to be autonomous tumor suppressor. Because if you go look outside, some representative function is SAHF, senescence-associated secretory phenotype. Senescence cells secrete so many different things, right? So it has a big impact on the microenvironment, tissue microenvironment. So the chronic inflammation, for example, like secrete a lot of cytokines, IL-1, IL-6, IL-7, 8, and the chronic inflammatory environment is actually tumorigenic most of the time, right? So non-autonomously often tumorigenic. And as now it's suggested already, it has a big impact in the longer term in our life, aging. So it's kind of not just straightforward, just describe this is tumor suppressor or tumor promoter. So the important thing, right? So if you look outside, like SAHF or whatever cells do something on the microenvironment, this is a functional cells which determine the cell type, right? To me, again, this is just me saying the pathological version of terminal differentiation. Right? It's quite similar in terms of the epigenome. So which means, right, so differentiation or cell type, what dictates cell types, which is lineage-specific genes. Lineage-specific genes often represent cellular functions. Here, this is Joanna. I just plotted this one. It's just I hand-picked few examples. a vertical axis, y-axis is a tissue specificity score. The higher, the more tissue specific, right? For example, insulin is the top, right? Only beta cell, almost only beta cell can produce. Other cell types silence insulin, right? Okay, so there's on the right secretome. This has the highest. This has also including a cell surface protein like CD3, CD4, CD8, right? So this has the highest tissue specificity. Of course, it's not surprising because we all use as a cell type marker, right? So this has the highest. And if you have look at the transcription factor, vast majority are quite ubiquitous. Some of them, the one you usually use for a reporter, right? Lineage reporter mice like FOXP3 and so on, except for those, the vast majority of transcription factors not really dictating tissue cell type. Cell cycle, obviously, right? They don't invent a new way to cycle, no? Apoptosis, even lower. So this is I like this analogy. This is the most, arguably not the most famous opening line of the literature. Apoptotic cells are all, cells die in the same way, almost, no offense, to the apoptosis. I got the claim from the apoptosis expert once. But I used to work on apoptosis. But senescent cells, right, senesce in diverse different ways. Right? The secretome can be different from the different tissue type of senescence, right? But we collectively call this a senescence or secretome that represent the heterogeneity of senescence. So in this aspect, okay, I don't need this. In this aspect, we have two main recent development. This one is the OIS spectrum, oncogene-induced senescence spectrum. So senescence is not just one thing like apoptosis, binary almost. Senescence is not, like a more continuous, like a trajectory. We just call them as, collectively call them senescence, but it's wrong, right? So now we identified, let's say if normal cells become senescence in an oncogenic stress context, what's in between, right? So there are lots of continuous trajectory of cell differentiation, so to speak. And actually, these guys is the one which could make cancer. This is largely focusing on the in vivo single cell approach. So now I'm not going to talk about this one today, but it's really, really important. And I expect that people change the way to see senescence, I hope. This is now in a Research Square preprint. It's currently under revision for the formal publication. But today's talk is focusing on the established in vitro model. This very model, ideal model system, right? And focusing on a high order chromatin structure. So the high C, I'm sure everybody knows about this one. As a high C, how do we do this? Right. So it's a long range interaction of the linear genome from here to here. How, if they bind to each other, you can identify by sequencing. Right. So then, doing the whole genome in the high depth. So now you can do the high resolution, high C map like this. So if this is one whole chromosome, let's say, or one arm, from this bit bind to here. So then this become redder. Blue means it's less than expected. So, and this is the typical picture you see, right? So this quite remarkable, actually, identify or confirm the long-term speculation of chromosome territory, and they can now divide it into the few, that's initially two A and B compartment, open and closed compartment. It's quite nicely separated. And then TAD, sub-megabase TAD, topological associated domain. This is more like a local connection, right? And then if you look closer, right, loop structure, which is from here is quite reasonably specific to tissue types. And then within this loop structure, loop domain, a lot of enhancer promoter interaction happens. So using this technique, so far we've been, I just highlighted 2 papers, recent papers. Joanna is sitting there, so both papers, Joanna is the main person who did all the analysis. So the fibroblast, we use fibroblast, human diploid fibroblast, oncogene-induced senescence. Fibroblast can activate, you can activate like SAHF-like genes, like cytokines, by just adding like a TNF or whatever. But nothing changed. Nothing happens to the enhancer promoter region. But in senescent cells, without any extra thing, senescent cells, look, if you look at the IL-1 locus, it looks like macrophage, professional cells, right? This kind of functional self shifts seems to be happening here. Senescence, you see the massive rewiring of enhancer promoter. But this is still euchromatic area, right? So if you look at the heterochromatic area, lineage, so how can I get rid of this? Oh. Try. Sorry, which one? Okay, that's going to minimize soon. So the lineage inappropriate gene activation. So many genes are highly heterochromatic and then they never be expected to be expressed. Right? In this example, just fibroblast again, start expressing the skin gene, only expressed terminally differentiated keratinocyte. This is keratinocyte, showing the similar loop structure. But the physical, actually this is by imaging, the physically heterochromatic region is disrupted, widely open, and then it starts showing this kind of loop structure. So it's, if you look at very high, this is gene-centric analysis, the high resolution, high C mapping, you can take a look at these kind of things. But it's still one by one, right? So currently, it's just going back to our original question. It's a more global picture, like a zoom out a little bit. How does it look like? So it's a nice introduction. Thank you very much. SAHF is kind of my life work. In the IMA90 human diploid fibroblast, if you overexpress oncogenic class in this senescence, but you see this kind of very striking heterochromatin formation. If you look closer, it's already mentioned, but you see this concentric layer structure. It's very striking, right? So that here, typically the K9 trimethylation, strong heterochromatin, constituted heterochromatin is perinuclear attached to the lamin B, lamins. But in senescence, lamin B1 is typically goes down, and then perinuclear heterochromatin, particularly green one, K9, is gone from here, right? And then move to here. So now this is layer structure. What it means, we don't know. But then this is actually one of my most favorite experiments still. It's quite a long time ago. But I just share with you why we think, what's the evidence for our conclusion. So the synchronization, this is before senescence, because senescent cells don't proliferate anymore. So you can synchronize cells, normal cells, and then label DNA replication by 2 different thymidine analog, right? So this, the EDU, BRDU can be stained in a different color. Once they are incorporated into the genome, then you can induce senescence. and then staying individually. This was one of the most striking data I had before. So the top one is just normal cells. You see the perinuclear heterochromatic thing, a little bit, and then this is early replicated genomic region, right? So the late replicated region, which is mostly heterochromatin, and this is the merged one. But after incorporation, you do the senescence induction. You see here. Late replicating region is going to the center or core surrounded by early replicating thing. Here, this is quite similar, K27, K9. This is a snapshot of current state, epigenetic state. This is the genomic history, same or similar. So this is the way, this is the current model, right? So the subformation is the spatial repositioning of pre-existing heterochromatin. Spreading happens, heterochromatin spreading happens at the embryo, but now this is the somatic example of how the next layer of heterochromatinization. But now, going back to the high-C analysis, now this is the typical way top, right? But Joanna, she's the hardcore mathematician with the strong background of graph theory and high-C analysis. Okay, why not using graph theory? So this is the way she's looking. The data looks like this, not like this. So the clustering in biology, we use many clustering methods. Almost all, if not almost, majority of them are trying to maximize modules, right? That's because that's intuitive. That's what our brain does. But actually, in a graph theory, it's a method doesn't do this. So the real-world network, there seems to be a modular type is not the majority, right? So it's a lot of different types of structure, like core periphery, for example. This is quite intuitive. Like if you have a core here, right, Chicago here, and suburb people is coming, commuting. Those people don't know each other, but they can be recognized as a single same community, and so on. So then we try, or she tries to identify, she doesn't ignore the hidden community. So this is just, I like this, just an example. I took this from the paper from there. So nowadays, everybody has a mobile. So it's so rich, like people moving around, like that kind of information. So this is a complex urban activity network in London. And the people are moving around, okay? So the individual node represents the customer, the business type, right? This is prior knowledge, okay? And then size represents how many times people are going back and forth. So, and then the color is the one computer determined, community detection. So what I found this is extremely interesting is pubs is larger than station. So just for the for defense of the Londoners, this is the nightlife, like 6 p.m. to 12 a.m., right? Not the day life. But I'm sure if we do this in Tokyo, you will see the big station, right? That's the Japanese city. I don't know in Chicago, but I would be interested what you think. But now it's interesting thing is coffee shop is here associated with the transportation rather than eat and drink, right? So it can be easy to explain because it's many coffee shop in the station. But this is the interpretation. Okay, so this is, but this still maximize the modular community, but I think it's maybe interesting to, you see the point. So now, this is the high-C data. Again, there's the top is the normal way, but based on the community detection or similarity of the binding, right? So this is now is a 200 kb bins. Each bin is in one node. So then reorganize based on the community detection or connectivity. So it looks like this. Right? The same chromosome is just simply switching around. And this is backbone visualization. This is another shocking data to me. I don't know if it's shocking for Joanna too. Right, so this is just color code gradation. Right, it looks really like this, no? It's just here. This is a heterochromatic marks. This is like almost like lysine 9, and then surrounded by catein. The same is far away a bit. So it's of course it's visualization is not the same and deformed, but it's conceptually quite similar. If you look, this happened to be OIS data, but if you look at growing normal cells, it looks pretty similar. Oh, I forgot to mention the very important thing in the replication timing. I don't have probably not time. So even in the normal cells, if you look closer, heterochromatin is surrounded by late replicating region surrounded by the early replicating region. So this kind of structure can be a fundamental structure. It's just senescence is kind of exaggerate this. So now, but this is so busy, no? It's computer. I think that's one of the reasons. It's a bit difficult to work, but this is generalized graph. That's the powerful bit of the graph. Now, individual node is not individual locus, loci, individual community, which is the triangle here. Right? Each community has a different genomic region. So now you can see here, so you can do many different things. So this is the Joanna's example on the British Railway network. So if you identify the most far away regions, Land's End and the John or Growth or whatever. Now if you define those pair, now individual nodes, they can have a unique coordinate. Right? The same thing here. So these guys are most far away, and then individual has unique coordinate. Now you can do the quantitative analysis or whatever you like. Right? This is quite striking. So she calls this chromatin gradient or network gradient. I can imagine that this property can be found in any different network, living or non-living. But anyway, so in this case, oncogene-induced senescent time course, you see the increasing of the gradient. Human ES cells differentiation, you see the similar thing. Right? If you look at cancer, this is all disrupted. This is all the genome-wide, all the chromosome average. But if you look at here, it's now individual chromosomes, and some chromosomes is even the other way around. It's opposite. Right? It's something going terribly wrong in cancer, some type of cancer. But what's the implication? So we're still struggling. Right? It's great to discuss with you guys. Joanna has a poster. She's now looking at the network vulnerability or resilience and so on. So it would be great if you. I'll just leave it to you. But one last piece of information here. So the cancer cells. This is one of the most established cancer cell therapy-induced senescence, and the lung cancer cell line. We can induce senescence-like phenotype in few cancer cell lines. You can see the restoration of this gradient. By the way, this is A549, this P53 intact. It's, the original gradient is relatively normal, so this might be the reason. But anyway, so this is kind of a snapshot of network, right? So individual description of the network. So now I'm moving to the next one. So the first one is just go to the poster. The second one also driven by Joanna, but now focusing on the differential chromatin maps. Not just one by one, differential. Okay, So this part is we just submitted, or it's already on a research square. Now it's currently under review. Oh, no, under revision now. Yes. So you can take a look at this in more details. Now I have to introduce another player. 5 minutes. Okay. Non-histone chromatin architecture protein HMG-A1. This is absolutely required for SAHF formation. And the literature or textbook says HMG-A1. This is the very ubiquitous chromatin protein. Okay, so there's no transcription factor or anything, right? But it can have a big impact on the structure and then gene regulation. And there's people been proposing the HMG-A1 can have an impact on the gene regulation in cis or trans. That's the prevailing model, right? Every review article is writing this. We don't have any evidence for this. Don't exclude this one yet completely, but we don't have a strong evidence to support this one. But anyway, so I come back to this point. So this is the high C data. If you look at the canine, that's a canine translation and HMG-A1 is a reasonably good correlation at this resolution. And then green, increased interaction, long-range interaction from canine to canine or HMG-A to HMG-A, right? And then when you, this part is not new, but when you knock down HMG-A1, it's largely disrupted, right? It's almost top green and bottom blue is kind of mirror image. This is great, right? This is kind of validating our imaging data. But it's still confirmatory, very expensive way to confirm what we've been saying. It's a little bit less insightful, if I may say. Okay, so this is the way. So Joanna wanted to do a completely different way of doing it. Again, this is differential, not the individual condition, right? So left is OIS minus growing. Okay, that's log forward interaction, connectivity, and the overlay with the HMG-A1 chip-seq. So you can see the quite big hub. The red edge is an increased interaction, but this is always or often associated, coupled to the blue edge is actually excluding here. So it's even HMG-A is low, this exclusion is dependent on HMG-A. Okay, so the canine translation is also good correlation, but not always. Right? So HMG-A seems to be more strongly correlated with those kind of aggregation. And HMG-A hairpin, again, many blue become red, vice versa. So it's largely dependent on the HMG-A. So that to analyze, to get some more information to introduce and implement K-core decomposition. This is just coreness. So some people have many degree. Right? Some PIs have many emails coming from your students or postdocs. But it's not necessarily good. Okay? If your student or connectee may also have a connector, have a higher degree, which makes you have a higher coreness. Right? There's a proper way to calculate. But here, its striking thing is the best correlation to the high coreness is HMG-A, even higher than canine translation. So I just go quickly. So the gene regulation. So you can, you can classify depending on this coreness. And one of the extreme one, the core, we call this is core regions, have those EMT genes or cell cycle genes. What it means? EMT means the fibrogenic, right? So that downregulation, largely downregulated in these genes, right? So they lose the fibrogenic activities. Again, the functional shift. And then ETF target, cell cycle genes shut down. The outside is excluded ones, all blue lines. These are mostly upregulated genes, are mostly SAHF genes, right? So it's not just changing the expression by you changing the global structure. Okay, global structure, that could be the key for the cell functional identity shift. I'll just go. Do I have one more minute? A few more, 2 minutes. So then, just looking at single cell data, because it's all average, the expression data. So this is this Western blotting showing oncogene-induced senescence with or without HMGA knocking down. It's relatively subtle. That's okay. That's the way. If you knock out, they just can't proliferate. And then if you knock down HMG1, the surprising thing is the SAHF genes even higher, right? That's what's the one. Is not nothing. Of course, it goes as you would expect. But if you look at here, this is single cell RNA-seq. This is growing senescence with or without HMG1 hairpin, right? Nice separation between these, but not much here, right? They are substantial overlap. But if you look at IL-8, the SAHF genes, you may see some correlation with this one and that, right? Purple. Now, if you only use the OIS bit, right? This is very powerful, like a differential abundant test. It's Milo, which was developed by John Marioni's group. They used to be with us, with our institute. So Milo can dissect these kind of things. So the Milo identified the 4 group of neighborhood. You first identify neighborhood, and in each neighborhood which is more representative, right? So those 2, one is OIS, more representative. RNAi is more abundant, for example. So now if you look at here, the SAHF gene is more upregulated in this cluster. But another hallmark of senescence upregulated here. So we used to think it's 1, 2. We never separate them out. But they seem to be more predominant in a different subset of cells. And these are, I would call them as a senescence or classical senescence. But 2 classic markers seems to be predominant in a different way. 3. If you knock down HMGA, SAHF is gone, but it's not really senescence, it's largely intact. But subset of them, actually, cell cycle genes are high, right? So then, I think this is incomplete senescence. So HMGA is still important for the senescence establishment. This is another unique one. This is the notch phase, which we previously identified. I don't have time to go into detail. But here, this is fibrogenic activity. SAHF activity is almost mutually exclusive. So this is a summary slide, final slide. Take-home message. So we don't have a strong evidence of this textbook thing. We think this HMGA has a big impact by making a chromatin environment. This further upregulates buffer activity probably could involve those things, but it's mostly inhibitory. This is very consistent with the idea of phase separation, which we only have evidence in vitro yet. But a take-home message, right? So HMGA1 may be as important as nucleosome or core histones, right? Another layer of gene regulation indirectly, not directly. Thank you very much. So I think I covered this one already. Happy to take your question.